I'd like to now introduce Dr. Ying Tam, who is the Chief Scientific Officer of Acuitas Pharmaceuticals, a company that he founded a few years ago together with Peter Cullis, another one of Peter Cullis's companies. And Ying has been very much involved in LNP formulations of RNA and other medical applications of those going forward. Thanks, Ying.
First of all, sorry, is that on? Yeah. First of all, I'd like to thank the organizers for the invitation. It's always fun, well, fun for me at least, to share some of the really exciting things that are happening at Acuitas. For those of you who don't know us, Acuitas is a biotechnology company. We're based in Vancouver, British Columbia, and our focus is exclusively on developing lipid nanoparticulate delivery systems to enable nucleic acid-based drugs. Somewhat unusually, we don't seek to develop our own drugs, but instead we work with multiple partners to apply our delivery technology to their payloads, and then we work together to move these medicines into the clinic and hopefully beyond. And certainly within that context, and as you heard from Peter, Acuitas is extremely proud of the contributions and the accomplishments that we've made. These include the development of a life-saving drug, Onpattro, which was developed in partnership with Alnylam Pharmaceuticals to treat an otherwise fatal disease known as transthyretin amyloidosis. And then more recently, work that Acuitas did with BioNTech and Pfizer to develop the COVID-19 vaccine Comirnaty, which represented in 2021 the world's first approved mRNA therapeutic and the first widely approved COVID-19 vaccine. Since then, Comirnaty has been administered over 4 billion times across the world and has been believed to have saved maybe in excess of 10 million lives. So certainly, we've seen a lot of success in that area. And while I could actually fill several of these talks with all of the additional work that we're doing to continue to evolve the vaccine technology, I thought what I would do, and for the remainder of the talk, is describe some of the work that Acuitas is doing to develop lipid nanoparticles for other types of therapeutics, nominally IV administered therapeutics, of which a number of the applications are shown here: protein replacement, delivery of therapeutic proteins, passive immunization, and I'll spend a little bit of extra time talking about the applications for gene editing. Now, Acuitas, one of the core pillars that has helped us continue to develop an effective lipid nanoparticles has been our novel lipid discovery program. And that contemplates, in an iterative sense, novel lipid. Our chemists imagine and design and synthesize novel lipids. Those get incorporated into lipid nanoparticles. They are then tested preclinically for safety, tolerability, and for potency. And all of those learnings are fed back into the next round of design. So we very much use a rational design approach to evolve our lipids. The success of that process is well demonstrated here. We call this the wave slide, but in essence, what each bar represents is a specific lipid nanoparticle. And this is results from an in vivo screening and on the reporting expression of a reporter molecule, luciferase in this instance. And so the only thing, and the composition, biophysical characteristics, all identical across these lipid nanoparticles. So the only thing that's different here in each of these is the identity of the ionizable lipid, giving us an idea of what that lipid contributes to the potency of the lipid nanoparticle. And of note, way at the far left-hand side of your looking for you guys, is MC3. That is the lipid that we developed for Alnylam to be in Onpattro. Still, by all accounts, a very active lipid. And nowadays, and this is somewhat of an older slide, we're talking about lipids that are, in essence, somewhere in the neighborhood of 25 to 30-fold more active than MC3. But in addition to just developing more potent lipids, we also put a lot of stock in understanding how exactly the lipids work in vivo. And the other thing which is very important in this instance is the fact that some people have said that mice lie, and so we recognize that a lot of the results we get in mice may not translate into human. So we do a lot of work in non-human primates based on the observations we made with Alnylam that, at least for the siRNA, LNPs, NHPs, very accurately predicted the activity of in humans. So we spend a lot of time evaluating our lipid nanoparticles in non-human primates. This slide shows the pharmacokinetic profile of 3 of our top lipids, and you can see that different classes of lipids can exhibit very different PK profiles, but what's interesting is that all of these lipids are highly active despite the significant difference in their PK profile. And the other thing which shown here is that the PK profiles are consistent across dose, low versus high, shown on this slide. In addition, the lipid nanoparticles perform equally well in male and female animals, shown on the left-hand side, and there's very little variability between individual animals, shown on the right-hand side. This slide shows 4 different lipid nanoparticles administered at a low and a high dose, at least for the first 3, and we can show that while there's there are nuances, that the biodistribution shows highest distribution to the liver, spleen, and adrenal glands, and whereas most other tissues are at low or below levels of detection. Something that people ask a lot, which is very important, is the ability to repeat dose. This is somewhat of a complicated experiment. In this, we're looking at 4 different lipid nanoparticles shown by the different colors of the bars at low and high dose, administered 3 times with a 1-month interval. On the first dose, we administered lipid nanoparticles with mRNA encoding immunoglobulin. In the second and the third dose, mRNA encoding alpha-galactosidase, and what you can see is the rank order, the pharmacodynamics, is extremely consistent across repeat dosing, with no impact on the level of expression. So we don't see a drop with repeat dosing. And importantly as well, which I know has been some stated concerns, is that if you look here, this is the pharmacokinetic profiles of the LNPs administered after the first and the last dose, and what you can see is that they're identical. So in this repeat dose scenario, there's no impact on the circulation lifetime. And also importantly is the fact that the tolerability, in this case, a readout by plasma transaminase levels, we can see that there's also no accelerating tolerability observations upon repeat dosing. And truthfully, the only impact is that the payload can show an impact, since that first dose was with a different mRNA. The final 2 doses are with the same mRNA, and you can see that the second and third dose, the ALT levels are very similar. So I just zoomed through that, and so here we are. We are armed with very potent lipid nanoparticles that have a reproducible pharmacodynamic activity, reproducible tolerability profile, and can be administered multiple times, and they behave and has reproducible pharmacokinetic and pharmacodynamic profiles. And so what does this mean? So armed with that, we have moved forward with a number of partners and collaborators to develop additional treatments in areas. This is an example of a collaboration with a group from the Boston University, looking at the potential use of the lipid nanoparticles to deliver, in this case, growth factors, hepatocyte and epidermal growth factors to treat liver disease. This shows a chronic liver disease induced by a choline-deficient diet, where in the upper left-hand panels, or in the left-hand side, the micrographs, you can see in the control LNP, denoted poly-C here, it's LNPs simply encapsulating poly-C RNA. You can see that the steatosis, the lipid accumulation in the liver, remains high over an 8-day post during the recovery period. Whereas if you would deliver an LNP with the 2 growth factors, you can see a reduction in the amount of lipid staining in the liver. And this is accompanied, shown on the right-hand side in the top panel, by an increase in cholesterol in the plasma as it gets released from the liver, and a decrease in liver transaminase levels, all indicating the ability to use this therapy as an example to treat chronic and acute liver disease. In the final few slides, I'm just going to talk about the application of our LNPs to gene editing. That's been a really an area that has generated a lot of interest, and we put a lot of work into it. There's a lot of advantages in regards to the use of the LNPs versus, say, viral delivery. Number one, there is very little limitation on payload size. And the other thing is that you can accommodate multiple payloads in a single LNP, which is important certainly when we're talking about CRISPR-Cas9 and the guide or the prime editing mRNAs that need to be included. As well, there's also the suggestion, there's enhanced safety profile. There's no prospects of insertional mutagenesis since it is an mRNA payload. And the fact that the mRNA engenders only transient expression of the editing enzyme leads to the expectation that there will be reduced off-target editing in those scenarios. And so one of the first, some of this slide just shows some early work we did with a couple of our partners, Sangamo and Bluebird, where we were able to deliver mRNA LNPs, which had mRNA encoding either a zinc finger nuclease or a TALEN editor, and we're able to show in the upper panel editing, in this case, of the transthyretin gene, the same gene that's targeted in L9 Onpattro. So you can see with two different ZFN sets, dose-dependent editing, and you get a corresponding reduction in the TTR, or transthyretin levels. In the lower panel, we see using TALENs from Bluebird that you can edit the PCSK9 gene, a gene associated with high incidence of cardiovascular disease. And so in the blue, you can see dose-dependent editing of the PCSK9 locus. In the red, you see a concomitant reduction in total cholesterol in the plasma. And in the right, you purely see the correlation between the two. And then a slightly more exotic application of gene editing is the potential to use the gene editor to cut the DNA and then hopefully insert a DNA into the cut site. And in this slide, you can see work done with Sangamo, again, where you're able to, with repeat dosing, 1, 2, and 3 doses, show elevated insertion of a donor DNA that was delivered by adenovirus or AAV, and then resulting in increased activity driven by the gene, which in this case is IDS, the inserted gene that was IDS. And then this is just work more recent from another partner, Precision Biosystems, that's proposing to use delivery of their ARCUS nuclease to treat chronic hepatitis. For those of you who don't know, for chronic hepatitis B, a lot of the persistence of the disease is due to integrated as well as covalently closed circular DNA. And the idea is that both in primary human hepatocytes and a non-human primate model, delivery of a ARCUS nuclease to target that hepatitis B DNA can effectively reduce the amount of residual hepatitis B genome in the cells, and ultimately leading to a potential cure. And finally, the last thing I'm going to talk about is work that we've done with Verve Therapeutics. Verve uses a somewhat more advanced editor. In this case, rather than just a cutter, it is a base editor. It's designed to treat or correct point mutations within diseases driven by a point mutation, because it's able to specifically alter a single mutation. In this case, they use the base editor not to actually correct that mutation, but to actually introduce a stop codon into the PCSK9 gene with the idea that if you silence the PCSK9 gene, which again is associated with a high incidence of cardiovascular disease, you can reduce the lifetime risk of that disease, or in people with high risk of cardiovascular disease, to reduce that risk. So administering an adenine-based editor for PCSK9, you can see on the far left-hand side that there is what is essentially is saturating editing at tolerated doses in non-human primates. That editing of about 70% in the whole liver results in about 100% reduction of PCSK9 and a concomitant reduction in about 70% in LDL cholesterol. Importantly, the editing is persistent, shown here, and this is an old slide, and I think we're out now past 2 years, and a persistent drop in the LDL cholesterol. So in summary, the systemic mRNA LNPs are suitable for therapeutic applications. They appear to be potent and well tolerated at clinically relevant doses. They're potent in primates, and they're compatible with repeat dosing. The mRNA LNP seems to be particularly suitable for gene editing applications, as shown by the data that I presented. And so with that, I'd just like to acknowledge everyone at Acuitas. I'm not going to tell you that this picture was taken in Vancouver, but in any case, this is the Acuitas team about a year ago, and as well as all of our collaborators and our partners. Thank you.